Hello there, my name is Dr. Amber Schmidt-Key, and this is a recorded lecture for my health science majors for Chapter 11 out of the Totora, Funky, and Case Microbiology and Introduction. It's from Pearson Publishing, the 12th edition. But again, this is a microbiology for health science majors class. And so we're going to get going. The purpose of this lecture is to talk to you about sort of a 20,000 foot view of the microbial world and the breadth and diversity of organisms that are there. So we'll obviously go into quite a bit more depth when we get to systems by systems infections. But for the time being, again, just want to try to impress upon you the diversity of the microbial world and how we organize a lot of our medically relevant pathogens. So it's important to recognize that we tend to think of microbes just in the sense of the ones that affect humans, but actually those organisms only comprise less than 5% of all the bacterial species that exist on this planet. In fact, the number of bacterial species actually outnumbers all the numbers of all other living things combined on Earth. So there is an enormous amount of diversity and an enormous amount of bacteria that will never cause you any harm. But for those that are medically relevant, we tend to organize those. We have several organisms that don't stain gram-positive or negative, that we have to use alternate stains. But we're going to talk primarily today about the gram-negatives and the gram-positives. So the gram-negatives are organized in a way that reflects a Greek god named Proteus. And Proteus was famous because he could change shapes. And so that's kind of the way that this name comes about. So that's how the gram-negatives are organized, and the gram-positives are divided based on their GC content of their genome. So if you remember from when we were talking about DNA, you know that A pairs with T and C pairs with G. Well, they pair because of hydrogen bonds. And there are more hydrogen bonds in a GC bond, there's 3 of them, as compared to an AT pairing, which has 2. And so they use this because the higher the GC content and the stronger those bonds, the more heat you have to apply to melt the genome into individual strands. So, of course, we'll go through this on a more individual basis when we get there. I don't want my students to panic. We are going to talk about a lot of organisms in here. What I want you to understand especially is how the organisms are divided and organized, less so than remembering that one is a proteobacteria versus one is a firmicute, that sort of thing. All right, so within the gram-negatives, the first major subcategory is called the proteobacteria, and this actually comprises the largest taxonomic group of bacteria for us. They were thought to have originated from a common photosynthetic ancestor. And the name proteobacteria, like I mentioned on the earlier slide, comes from this Greek god who you see sort of depicted in this image called Proteus. And here you can see he's kind of this chimera of a human and a sea serpent. And so that was kind of the idea. The group Proteobacteria is divided using Greek letters, since we're already talking in Greek. So it'll be alpha, beta, gamma, delta, and epsilon. So again, you don't have to remember which ones are beta and which ones are epsilon. The point is just understand how we organize the bacterial world. So with this group, it's based on Greek, and then there will be more information to come. So within the alpha proteobacteria, many of these are not going to be really relevant for us. Many of them are going to be important for agriculture. So they're used for, these organisms can do nitrogen fixation, which means kind of like photosynthesis, which fixes carbon dioxide into organic materials. They can fix nitrogen into organic materials. But they will also cause disease in plants. They're some of our plant pathogens. On the human side, these pathogens will tend to be intracellular parasites, meaning that they're not going to do well outside of a human host cell or another animal reservoir host cell. So the first up is rickettsia, and we'll talk about these later on in the semester. But these are vector-borne, and that just means that they're transmitted using an insect, especially a tick. They tend to gain entry into the host cells by tricking the host into phagocytosing them. And they appear as a rod shape or coccobacilli under microscopy. So the diseases that are most often associated with the rickettsias are the spotted fevers, including typhus and Rocky Mountain spotted fever. So stay tuned. We'll be talking about these again later in the semester when we get to systems by systems infections. Ehrlichia is the next one up. It's a lot like rickettsia in terms of being tick transmitted, and it's still an intracellular parasite. In fact, these ones will live inside of white blood cells. And so the disease it causes, ehrlichiosis, will give flu-like symptoms, which can make it a little bit hard to spot and hard to diagnose unless you are looking for it. Bartonella henselae is the next one. It's a bacillus, and it causes cat scratch fever. So this is an infection that results from a wound, often from a cat scrape or scratch. Brucella is a small non-motile coccobacilli, so it cannot move. And this one relies on zoonotic transfer, meaning contact with an animal. And that does not necessarily mean ingestion. It can mean working with the animal, working with their hide, that sort of thing. And so in humans, it's not that big of a deal. Brucellosis can cause flu-like symptoms, but in cattle, it can cause spontaneous abortion. And so that's clearly a problem in the veterinary community. The next group is called the beta proteobacteria. And so the first organism here is Burkholderia. This is a motile aerobic rod-shaped organism. The first organism we're going to talk about is Burkholderia cepacia. This is an interesting organism because it can eat a lot of different things, and it's an important cause of hospital-acquired infections, and it is sort of the kiss of death for people who are affected by cystic fibrosis. So cystic fibrosis patients tend to have some sort of a lung infection for most of their lifespan because of decreased or increased mucus, dehydrated mucus that inhibits mucociliary clearance. And so they'll have this fertile medium, the mucus in their lungs, where bacteria can grow rather happily. And so they'll be infected with a certain population of organisms for a long time. But the problem is once Burkholderia cepacia takes hold, that usually, sort of forecasts a decline. And it's sort of the beginning of the end. So it's important. Burkholderia pseudomallei is a tropical disease, and it's considered a potential bioweapon, but we'll talk about that more later in the semester. Bordetella has two interesting organisms. It's an aerobic rod, and for the Bordetella pertussis, this is what causes whooping cough. This is a non-motile vaccine-preventable disease. So we'll be talking about that in respiratory infections. And then Bordetella bronchoseptica is pictured over here on the upper right. This is a motile organism, and it's the one that causes kennel cough. So if you've ever had to board one of your animals at a boarding facility, they'll often require that you have a kennel cough vaccine, and this is what that is. Neisseria is another really interesting set of organisms. They appear as diplococci when you look at them under the microscope. And they are aerobes, so they will tend to inhabit mucous membranes that have plenty of access to air. So the first one up for us is Neisseria gonorrhoeae, which causes gonorrhea, the sexually transmitted infection. And then the other one of interest is Neisseria meningitidis. This causes meningococcal meningitis. We have various kinds of meningitis, but this one's called meningococcal after the name. But we have a lot of asymptomatic carriers. In fact, we have about 20% of the human population is estimated to be an asymptomatic carrier of this. Within the gamma proteobacteria, so we're still in this larger group of the proteobacteria, this is actually the largest subgroup within that. The first is the Thiotrichales, and this is best represented by the organism Francisella tularensis. This is a small pleomorphic organism that just means that it can adopt multiple shapes. And it requires enriched media. So it's a little bit of a needle in the haystack when you're trying to find it. And you sometimes have to really encourage its growth in order to detect it. The disease it causes is called tularemia, which is also known as rabbit fever. It is considered a potential bioweapon. It's usually transmitted through ticks, so it's a vector-borne disease. And you'll notice when we get to it that it causes very enlarged lymph nodes. The next grouping is the Pseudomonales, which includes rods or cocci. So Pseudomonas is depicted up here in the right. We have two cells here. Looks like one is maybe preparing for cell division, but they're motile by flagella. They're often pigmented, so when you grow them, they'll appear blue or green sometimes. And they have a very familiar sort of or characteristic grape-like odor. It smells like Dimetapp cough syrup. So one of the most notorious of this group is called Pseudomonas aeruginosa. It is considered like the ultimate opportunistic nosocomial pathogen. Opportunistic means it's just waiting for a chance to make a move. Nosocomial means hospital acquired. So this guy is just waiting in the wings, looking for any way to get inside of a human body. And so this can range from UTIs, especially somebody that's had a catheter, because this is a super biofilm former. It also can cause problems in burn wounds. What tragically we used to do with burn victims to provide some relief to them is that we would mist water over, like a spray bottle, over their wound site, which provides some temporary relief because of the cooling sensation. But unfortunately, Pseudomonas aeruginosa lives quite happily in water. So it meant that we were seeding an infection into these people who were already super vulnerable with their skin no longer intact, and they could lead to life-threatening infections. One of those life-threatening infections is called sepsis. We'll talk about this a lot later in the semester when we get to, well, actually, I think it's just a couple lectures from now when we talk about pathogenesis. Cystic fibrosis is another, I mentioned Burkholderia cepacia earlier. Pseudomonas is the organism that most often colonizes cystic fibrosis patient lungs, and it's with them for a very long time. And it can be very difficult to treat because it is a biofilm former. It tends to be antibiotic resistant, and it can also be antiseptic resistant. And it can virtually grow anywhere. So pretty problematic. The next organism is called, hold on just a minute. Okay, sorry about that. Hopefully the dog problem is on its way to being completed. I apologize for that interruption. The next organism is called Moraxella lacunata, and this will cause a conjunctivitis or eye infection, and it usually will appear as a coccobacillus. The final organism within this group that we're going to talk about is called Acinetobacter baumannii. And this is a soil or water organism. It tends to be very antibiotic resistant because it is a biofilm former. And it is considered an opportunistic nosocomial or hospital-acquired infection. And in the last couple decades, it's become more and more prevalent and interesting because it's associated with battle wound infections, especially in Iraq and Afghanistan. So what would happen is it's endemic in the soil there, and we would have situations in battle where there would be like an IED blast or other sort of explosion that could cause wounds, serious wounds. And so they would try to medevac them out using a helicopter. And when the helicopter came to land, the rotor wash would stir up so much dust and soil that soil and dust, including the Acinetobacter baumannii, would land in the wound, seeding that infection into very fertile tissue. And so it can cause life-threatening infections, including or other complications like amputation and other problems. It's very hard to get rid of once you have it, so that's another complicating factor. All right, so the next group is called, let's see. Why aren't we moving? Okay, the next group is called, we're still in the gamma proteobacteria. This will have the Legionella here. And so Legionella pneumophila is probably the one that's most recognizable from this. It causes Legionnaire's disease, which was a interesting outbreak in the 1970s at a conference of the American Legion, excuse me. So that's where the name comes from. And this causes kind of a pneumonia. And so at this convention, you had all of these older gentlemen attending, and a couple days to weeks later, we had all of these spikes in cases of pneumonia. And they started to put it together that these cases must be related. And so they did an investigation of the hotel where the event was held, and they found that this organism, Legionella pneumophila, was living in the cooling towers of their HVAC system. And so just sitting there in the ballroom or the conference room listening to presentations or participating, they were inadvertently breathing in this pathogen. So in modern times, this is definitely a concern in water sort of repositories. So HVAC systems, the water lines of hospitals, and misting equipment. So that can include things like the produce misters at the grocery store. It can also include like cooling stations, misting sort of fans that you see sometimes at amusement parks. In fact, Disneyland had an outbreak associated with one of their misters of this disease a couple years ago. Coxiella burnettii is another organism within this group. It is an obligate, meaning it has to be an intracellular pathogen, and it causes something called Q fever that has flu-like symptoms, which may seem a little bit minor, but it's actually considered a potential bioweapon because while flu-like symptoms doesn't seem that bad, if it's affecting a wide swath of the population, then community functions start to come to a halt. And it can cause a panic is the other thing. So it's ordinarily transmitted by contaminated milk, but that likely would not be the case in the event of a biological attack. The next family is called the Vibrio, and this group of organisms will be facultative anaerobes. They tend to be curved rods, looking a little bit like a cheese puff in shape. When you look at them under the microscope, you'll notice these strings attached to the ends. Those are polar flagella, so this is definitely a motile organism, and that's important because they tend to live in water, so they need to be able to move in that environment. There's 3 organisms we want to discuss here. Vibrio cholerae is the most important, and this one causes cholera, which is a watery diarrhea that can be fatal in less than a day. It's transmitted through a fecal-oral route of transmission, and it is often through the ingestion of contaminated water. Vibrio parahemolyticus causes a less severe gastroenteritis than Vibrio cholerae, and it's associated with shellfish. Vibrio vulnificans is another kind of infection that causes wound infections, and especially because this organism will grow in the shells of oysters, clams, and other shellfish. So when you walk along the beach and barefoot and step on a shell, and if it cuts you, there's a risk for this sort of infection, and it can be rather nasty. The largest group within, or maybe not the largest group, but a very important family within the gamma proteobacteria that I do want you to know is the Enterobacteriaceae. It's the only family I really need you to know, and they're called the enterics. You'll notice that you know many of these organisms already. So they use fimbriae to attach to mucosal linings and then cause infection. They're transmitted usually by a fecal-oral route. The exception to this is Yersinia, which causes plague, Yersinia pestis. That one is transmitted through a vector-borne route and sometimes a respiratory droplet transmission, so when somebody coughs or sneezes. Organisms within this family include Escherichia or E. coli, for example, Salmonella, Shigella, which causes dysentery, Klebsiella, Enterobacter, Serratia, and Proteus. Several of these we've already worked with in the laboratory. Pasteurella is another organism within the gamma proteobacteria. They are non-motile, and they represent both human as well as animal pathogens. Pasteurella multicida is an organism that can inhabit the mouths of dogs and cats. So the way we get these infections is through their bites. Remember that these bites, and especially from cats who have narrower but sharper teeth sometimes, are able to puncture deep within the skin, and that's an effective way to transfer Pasteurella multisida. So when there is an animal bite, it's important to wash the wound right away, but also to seek treatment for antibiotics. The next infection in our lineup is Haemophilus, or organism, is Haemophilus influenza. So the name here, influenza, is a little bit of a misnomer. It is the organism's name, but this is not influenza like influenza that you get a flu shot for. Haemophilus is Latin, where philus means loving and hemo means heme, like hemoglobin. And so when you put those things together, this is an organism that loves blood, or loves red blood cells in particular. They tend to attach to mucous membranes, and we used to think it was actually the etiologic agent of influenza, which is why it has this name. But in reality, it's really more of a secondary infection where you have true influenza first, and that weakens your natural defenses and activates your immune system and sort of distracts it from an additional threat, which is when Haemophilus influenza comes on the scene as a secondary infection and can gain a foothold. It does require blood for growth in culture media, and that makes sense based on the name. We're telling you it's Haemophilus, so it loves blood. But the reason for that is because, excuse me, these organisms are unable to make the components of the electron transport chain. So some of the diseases that are associated with this include childhood meningitis, which is why we have a vaccine. It can also cause earaches, bronchitis, and pneumonia. Then to round out the proteobacteria, we have the delta proteobacteria, and they are predators of other bacteria. So while that is super cool, it is not medically relevant. So we're going to go ahead and skip that for the time being. But feel free to Google that and learn about that through self-study. The epsilon proteobacteria are going to be rods that are helical or curved. They are motile with flagella. There are two organisms within this that we want to talk about. There's Campylobacter jejuni, which is the leading cause of foodborne disease outbreaks. Often you hear E. coli and Salmonella getting a bad rap, but actually this is the number one. And so it tends to be associated with poultry products. So that could be chicken or turkey or quail, any kind of fowl. They are microaerophiles, so they do require a little bit of oxygen for sure, but they don't want normal atmospheric oxygen. So they will live under the surface of a broth tube, for example. And then lastly, we have Helicobacter pylori, which is an organism that you should have learned about through doing the TED Ed talk assignment that I gave you, and you turned it in. So this is the most common cause of peptic ulcers and stomach cancer. And this story is notable because the way that the link was made between the organism and the disease. So this is Barry Marshall and Robin Warren. They're Australian microbiologists and pathologists. And we used to think that stomach ulcers were mainly due to stress and the consumption of spicy foods. And in 1982, these two gentlemen used a very unconventional way of demonstrating the link between Helicobacter pylori and peptic ulcers and stomach cancer, where Marshall, the gentleman on the left, drank a broth tube that contained Helicobacter pylori because nobody was believing him, and so he drank it, which is something we have always warned you not to do in the laboratory. He ended up with gastritis, which kind of proved the point that this was, in fact, the etiology. Thankfully, that's something they can cure with antibiotics. So they went on in 2005, a little over 20 years later, to win the Nobel Prize for this discovery. As it turned out, and the reason why is because it wasn't just a cure for gastritis, it wasn't just a cure for peptic ulcers, but actually Helicobacter pylori is a significant cause of stomach cancer. And so in a way, we sort of cured cancer here because all it took was an antibiotic, and now we've reduced a significant cause of cancer, especially in the developing world. All right, so now we'll talk about the non-proteobacteria gram-negative organisms, starting with the Chlamydiae. So there's 3 organisms within this family, and so first let's talk about let's go ahead and talk about them first. The most famous of these is Chlamydia trachomatis. This is what causes the sexually transmitted infection, a urethritis, where there's inflammation of the urethra. There's also chlamydia psittaci, which is a respiratory infection. And then there's chlamydia pneumoniae, which causes, as the name implies, a mild form of pneumonia. So these are obligate intracellular parasites. They really need to be inside to be fully capable of what they need to do. So in fact, when they enter a cell, they enter not as a real cell, but as what's called an elementary body, which is almost like a spore, but it's not quite a spore. And so they enter the cell, getting in, and then once they're in there in a vacuole, this is where it starts to reform its normal self. And so we have a cell now within that vacuole, and it's called a reticulate body at this point. So many times over, this reticulate body will divide inside of that vacuole, and then eventually those cells or reticulate bodies will start to generate new elementary bodies. It's very much like the sporulation idea, but it's happening inside of a eukaryotic cell. Those elementary bodies will be released from the cell to carry on the cycle. And so you can see this on electron micrograph where we have an elementary body here, and then inside of a eukaryotic cell, you'll start to see reticulate bodies inside of that. Okay, so still carrying on through the non-proteobacteria gram-negatives is the organism Bacteroides, excuse me. And these are obligate anaerobes, so they do not want the presence of air. They tend to be non-motile, and they tend to cause infections due to puncture wounds or surgery. So as it turns out, even though the gut has a lot of air passing through it, through what you inhale, swallow, etc., there's about 1 billion cells of Bacteroides fragilis in a gram of feces. So it's very, very prevalent in the gut, and it can cause peritonitis or perforated bowel. You also tend to have it in your mouth, and you may not think of your mouth as an anaerobic environment, but you actually do sit with your mouth closed and breathe through your nose for most of the day. And so it is actually an anaerobic environment when you're not talking or eating. And so they tend to live in the gingival crevices, and this is what can cause periodontal disease. The next thing for us is Fusobacterium, and these are interesting because they have this really interesting morphology where they look like little matchsticks or toothpicks. They have cells with very pointed ends, but they are known to cause dental abscesses and can also be found in the gastrointestinal tract. Lastly, we'll talk about the spirochetes, These are motile organisms using axial filaments. So it's not a true flagella. What's happening here is they have an embedded flagella in their cell membranes. And so this is how they move through like a corkscrew sort of movement. These are found in the oral cavity sometimes, and that's actually one of the things that Anton van Leeuwenhoek saw when he did those teeth scrapings looking at his rudimentary microscope. So some of the organisms that fall into this category include Treponema pallidum, which causes syphilis, a sexually transmitted infection, and Borrelia burgdorferi, which causes Lyme disease. Lyme disease is transmitted through ticks. It's a vector-borne illness, and we'll talk about it some more when we get to the system-by-system infections. The next organism for us, or we're going to shift gears at this point and talk about the gram-positives. These organisms are based on, or they're going to be organized rather, based on the GC content of their genome. So we'll have two subdivisions within this. We have what's called the firmicutes, which have a low GC content, and there will be another group of organisms that we'll talk about going forward in a little bit. So the firmicutes, if you think of the word acute, like an acute angle, means that it's small. But also think about babies. They're cute and they're small. So if that helps, go with it. If it doesn't, then pitch the idea and keep going. But these organisms have a low GC content. They're cute, they're small. So it's between 21 and 54%. Among these, we have some really important endospore-forming organisms. And some of the most important of these include Clostridium, which are going to be obligate anaerobes who do not want to be in the presence of oxygen. And so uh, notable organisms within this include Clostridium tetani, which causes tetanus, which is vaccine preventable, thank goodness. Botulinum, which causes botulism. Clostridium difficile is an opportunistic infection that's associated with antibiotic use. So I actually have a friend who, while she was pregnant, when you're approaching delivery and you're in your third trimester, you'll tend to, during one of your prenatal visits, they will screen you for the presence of group B strep in your stool through a rectal swab usually. And if that comes back positive, they will typically administer antibiotics during delivery to prevent the transfer of group B strep to the baby during delivery, because it can cause significant problems with meningitis and other things. And so she was positive and she was given antibiotics during birth. And during her postpartum recovery at home, I think the baby was no more than like 3 or 4 weeks old, she came down with a severe diarrhea sort of illness. And what it ended up being was this Clostridium difficile. And, you know, when she called me to ask about it, I asked, were you positive for group B strep during your prenatal visits? And she was like, how did you know? And it's because I knew they would have given her antibiotics if she was positive. So this is what Clostridium difficile looks like. And it is an important infection and becoming more and more important in the hospital environment. Clostridium perfringens is associated with gas gangrene and can cause a foodborne illness too. The next thing is bacillus. So bacillus they're typically soil organisms. So Bacillus anthracis is one of them that includes anthrax, or that is anthrax, and this is considered a bioweapon, but ordinarily it's a zoonotic infection. So you get it from working with contaminated animals or their products like hides or other things. And then Bacillus cereus is an environmental pathogen or environmental organism that can sometimes be implicated in foodborne disease because it happens to, the soil will contaminate water sometimes, especially in rice production. So if you're unfamiliar with how rice is grown, they tend to inundate the fields either naturally through flooding or intentionally in order to grow the rice. And so the soil that underlies this space will have bacillus spores in it. And so this can float in the water and contaminate the rice. So when we look at this image, you can see the spore. It's sort of this knotted space inside of a larger bacillus. But I don't want you to get the impression that all bacillus organisms are bad. Bacillus subtilis is something we work with across the hall, and it can cause infections, but it's a little bit more of a safe for us to work with BSL-1 sort of pathogen or organism. The next group within the firmicutes is our non-spore formers. So the first of this is going to include Staphylococcus. They will appear, as the name tells you, like grape-like clusters under the microscope. And the most famous of this is Staph aureus, which will appear yellow on the agar. If you remember from chemistry, the periodic table, the abbreviation for gold is AU. And that's because when you grow this on a blood agar plate or many different kinds of agar, the organism grows with this pretty golden pigment. And so that's where it gets its name. They are facultative anaerobes, meaning they can live in reduced oxygen conditions and carry on using things like fermentation. We have a lot of people who are asymptomatically carrying this in their nostrils. And it's actually, a lot of the times people forget about this, but it's actually a common foodborne pathogen, especially in foods that contain a lot of salt and a lot of sugar, sometimes because they're able to withstand high osmotic pressure, meaning that these guys are the ones that like high salt content, or they can survive that. So there is a risk. Organism can produce several toxins that will help with its pathogenesis. We'll talk about that later in the semester. It's also known as a nosocomial pathogen, meaning it can cause infections in a clinical environment, and it is increasingly resistant to antibiotics. Excuse me. So if you've heard of MRSA, that is this. It's methicillin-resistant Staph aureus is what that stands for. So not all Staph aureus is going to be MRSA, but there's a lot of MRSA out there, and it's becoming a growing concern. An infection we don't see quite as much anymore is toxic shock syndrome associated with Staph aureus. This was due to prolonged inappropriate use of tampons and feminine hygiene products, but as education efforts increase to try to get the word out about this, it has diminished, and it's not as prevalent of an issue as it used to be. The last organism we're going to talk about here is the lactobacillus, including lactobacillus. This is an organism that gets its name because of its shape and its ability to produce lactic acid through fermentation. So it is a natural colonizer of the vagina, the intestinal tract, as well as the oral cavity, and it is also what you use to make yogurt. So, fun fact. The next group within the lactobacillus includes Streptococcus, which we talked about just a minute ago in the context of Clostridium difficile. There's many pathogens within this genera, and they're mostly known for their ability to produce certain toxins. And so we organize the Streptococci based on their ability to carry out what's called hemolysis, or lysine red blood cells. So this is a blood agar plate over here, and what you'll see is that the agar is usually this dense red color, and then you'll have the colonies, and you'll notice that there's a zone of clearing throughout the media from all the way to the plastic surrounding these colonies, and that's because they're able to lyse or break the red blood cells that are surrounding them. So this is called beta hemolysis when you see it. And so organisms that will fall within this category include the group A strep, which is called Strep pyogenes. This is the etiologic agent of scarlet fever, pharyngitis or strep throat, erysipelas, which is a skin infection, as well as impetigo and rheumatic fever. You can also have Streptococcus agalactiae, also known as group B strep, which can cause neonatal sepsis, meningitis, those kinds of complications. Very serious, which is why that story about my friend having the baby was relevant. Then we also have the non-beta hemolytic strep. So they will not necessarily do this kind of hemolysis. What they will do instead is called alpha hemolysis, where, do I have a picture of this? No, I don't. Where instead of seeing this clearing, you will see a green ring around the colonies, and that is called alpha hemolysis. It's pretty distinctive when it happens. Organisms within this category are pretty important too. Streptococcus pneumoniae causes pneumonia, ear infections, that sort of thing, and it is a vaccine preventable disease. The trade name for this is Prevnar, and they go after like the top 23 strains of Pseudomonas pneumoniae that are common out there. But we've seen some evolution in the population around that vaccine, so that once you kill off those top 23, new strains take prominence. And so we've seen a little bit less effectiveness of this over time, and potentially a redesign going forward in the future. Then the next one is Streptococcus mutans, which is known to cause dental cavities. So this is what a Streptococcus looks like under the microscope, a chain of circles. Still within the same family, the Lactobacillus is Enterococcus, which is going to be an organism that is very well adapted to areas that are rich in nutrients but low in oxygen. So they will probably enjoy spaces like the GI tract, the vagina, the oral cavity, and this is a biofilm former. It's known as a fairly concerning contaminant in hospitals where it is a nosocomial pathogen, and it's especially alarming because it tends to be resistant to many antibiotics. In fact, just like we have MRSA for Staph aureus, we have another name or another organism with a similar kind of abbreviated name, and it's VRE, which stands for vancomycin resistant enterococci. And what's kind of alarming about that is vancomycin is a drug of last resort. It's a drug we don't use very often. And so for this one to be the one that they're resistant to, tends to mean that these are multi-drug resistant pathogens and we're running out of treatment options. So organisms that fall within the enterococcus include Enterococcus faecalis, which we do work with across the hall, and Enterococcus faecium. So the name sort of makes you think of feces, and that helps to remind you where they can be found. Another organism within this group is the Listeria monocytogenes, which is known to be a psychrophile. It can do fine in refrigerator temperatures, and so it's known to contaminate dairy products. And for example, when you're pregnant, you're not supposed to be eating deli meats or soft cheeses. This is the organism. Why? Because it can cause spontaneous abortion, preterm delivery, low birth weight, and other birth defects. It's considered a foodborne pathogen. You do consume it through your food. And we've had outbreaks in cheeses. We've had an outbreak in cantaloupe while I was working at CDC in Atlanta. So it can infect a lot of or contaminate rather a lot of foods that you don't necessarily cook before you eat them, and that's the biggest risk, honestly. Once it is ingested, what happens is it will live inside of a cell that has swallowed it. So it is an intracellular organism. It very rarely even leaves the shelter of a cell, making it hard for the immune system to find it and get rid of it. The next group is the Mycoplasmatales, which are highly pleomorphic, meaning they adopt many different shapes. They do not have a cell wall, and they are very, very, very small. In fact, it's the smallest genome of a living thing we know of, containing 517 genes. So it's almost akin to a virus, except that it does have a cell membrane and carries out a lot of cell functions. The organism that falls within this is Mycoplasma pneumoniae. It causes a mild form of pneumonia, and it tends to grow better in cell culture than using traditional media because it is an obligate intracellular parasite. From there, we'll switch from the firmicutes to the actinobacteria within the gram-positives. These are going to have a higher GC content in their genome, some as high as 73%. One of the most important within this group is the Mycobacterium. These are aerobic organisms that are rods with no endospores. They are called Mycobacterium because they have a mycolic acid cell wall. So because of that, we can't actually gram stain these. We have to use something called acid-fast staining instead. These organisms are very resistant to drying, to antibiotics, and they are notorious for having slow nutrient transport, which corresponds to a very slow growth rate. So it can take a very long time from initial exposure to symptoms because of a lengthy incubation period, but it also complicates diagnosis and treatment because we want to culture this and it can take up to several weeks for us to get a positive. So organisms that fall within this class include Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. So we'll talk about both of these when we get to the systems infections. The last one I want to talk about on this slide is Corynebacterium diphtheriae. So this is what it looks like. It's got a very sort of distinctive club shape appearance, but otherwise it's pleomorphic, and you can see that even from the smear. Some of them will look like clubs, some of them just look like 3 dots tied together, but this causes the disease diphtheria, which you've probably never seen and hopefully never will, because it is a vaccine preventable disease, and as long as we maintain effective vaccination rates in our population, this is more or less a developing world problem. We don't really see this in the United States. Let's round this out. We have the Propionibacterium acnes, which is something we'll talk about again when we get to skin infections, because this one causes skin acne. It is named Propionibacterium because it forms propionic acid. which is why if you've ever, I don't know, popped a pimple and smelled the stuff that comes out of it, it will have sort of an acrid smell to it. That's what you're smelling is the propionic acid. Gardnerella vaginalis is, as the name sort of tells you, causes a vaginal infection called vaginitis. Anytime you see the word or the suffix itis, that means inflammation of the root term that comes ahead of it. So this is inflammation of the vagina. Um, and so we'll talk about Gardnerella vaginalis later on when we get to the reproductive system infections. So that leads us to some review. There is a Kahoot for this. If you're friends with me on Kahoot, we can review there. Otherwise, we'll start the next lecture when I can be there with you in person with that Kahoot to make sure everybody kind of understood what we went through here in this lecture. But do try to work on identifying diseases as you thumb through the notes that were vector-borne, vaccine preventable. If they're a foodborne pathogen, that's really interesting. If they're nosocomial, antibiotic resistant, or biofilm formers. Again, I don't necessarily need you to remember that a certain organism is an alpha proteobacterium, but what I do want you to remember is how generally the gram-positive and the gram-negative organisms are organized. So on the gram-positive side, it's based on GC content. For the gram-negatives, it's based on these Greek sort of prefixes. And that concludes our presentation. Thank you.